So tonight I want to focus on a topic that's dear to me because it's the area that I work in, and that is the topic of omics. And omics is kind of a broad term that refers to a detailed or comprehensive analysis of basically any biological system. When we say system, sometimes it sounds confusing, it sounds very mechanical. What we just mean is very broadly, it could be something as large as a whole organism, like a whole human, or it could be something like just a heart. So a system could be any variance in between that, right? So a whole entire organism, or maybe just a cell in an organism. Anything that works as a system in biology. And the area that this name came from, omics, is from genomes. So you probably are familiar with the term, at least genomics. And that's because this is where we coined the term. When they did the sequencing of the human genome, we ushered in this whole area of systems biology, or again, just where we're focusing on a whole comprehensive analysis of a system. And that's where the omics technology came from. But we can now, and we do use the word omics away from just genes, we can use it to just generally talk about the collective use of anything. So, for example, anything that ends in omics is just the study in biology of the whole system of that thing. So, genomics is all of the genes. There's also metabolomics or proteomics. So again, it just is any of those fields respectively where you're studying the whole data of that particular system or organism. So we're going to talk about these four main omics. There's actually a lot of omics. They start getting very specific. There's even things called like nutritional omics or toxicoomics. They start getting very, very, very specific. And now that this field is out there, there's more and more and more terminology that's coming with it. But these are the four main omics that you will see, and most of the omics are related to these. And each of them is related to a particular kind of area of that central dogma that we talked about in biology. So there's genomics, again, which is the study of the complete set of genes or DNA in an organism. There is transcriptomics, which again is going to be the study of a complete set of RNA in any system. And here it's not RNA omics, right? Genomics is genomics. Remember that this process of DNA to RNA is called transcription, and that's where we get that transcriptomics. The study of all of the proteins in a system would be called proteomics. And then the very last one, and this is the one probably people are not as familiar with, is the study of all small molecules in a system. So this can be broadly classified as literally anything that is a small molecule and exists in a biological system. So anything that's not a protein, that's not RNA, and that's not a gene, and it's small. It could be a neurotransmitter, it could be a sugar, it could be anything. That all falls under the umbrella of metabolomics. So we're studying metabolites. So first, let's look at genomics. Again, genomics is the study of all the genome of an organism, and the genome is an organism's complete set of DNA. That's all of its genes. So this is in contrast to something like genetics. If you ever hear the term genetics, this is where we're looking at the study of individual genes. So we might be focusing on one gene and understanding that gene and their roles in inheritance. So if you've taken a genetics class, you probably have talked about how a particular gene influences something like eye color, and how that is related in your genetics, right? How that can be passed down. Again, genomics, in contrast, is where you're taking an organism and you're profiling all of the genes that it has, and you're looking at those genes. The areas that the topics this aims to get at in genomics is you get to characterize and quantify an organism's genes. And it's going to give you information about those genes interrelations and the influence on whatever organism you're looking at. So if you're looking at a human, you might classify all of its genes and then say, oh, this gene looks different than another person's gene. And so we know that this gene influences certain diseases. So generally, in order to compare DNA in genomics, we have to already have a genome map, right? We have to have an established set of DNA that we know exists to then compare the DNA that we collect from a person or from an animal or from a plant to. So this just kind of highlights one of the importances of the Human Genome Project. The whole thing that sparked genomics was we first mapped out the human genome. We know all of the genes that exist in their potentiality in a human, and then we can compare any of the DNA that we collect from a human to that map. So genomics needs to have this kind of already established roadmap in place for you to then compare to. One of the main uses of genomics is to kind of understand diversity at a whole genome level. And this is going to give you insight into things like traits and disease susceptibility. Generally, you're comparing. So you're taking a group of people who maybe have a disease, for example, and you're profiling all of their genes, right? So you're going to collect all the genes from all the people who have a disease. And then you might collect all the genes from people who don't have a disease. And what you would then do is compare. You'd say, I have all the genes from these people that have a disease and all the genes from the people that don't have a disease. What is the difference between them? And you would find maybe, oh, hey, there's a genomic variant between the people that have a disease and the people that don't have the disease. And this is a way that we can tell genomic susceptibility, for example, the potential susceptibility to get a disease if you have a particular variant. This is also how we can kind of highlight specific traits, right? Phenotypic traits that exist, for example, things that we express phenotypically would also be able to be categorized using genomics if you did comparison of people who have or do not have a trait. Now, genomics is static. This is the one thing about it. You're just getting that person's DNA. You're not, for example, the person might not have a disease, but they might have the potential to get a disease. All you have is their set of DNA. You don't know what's going on phenotypically, what's being expressed in their body. For example, just having someone's DNA doesn't necessarily mean that they have cancer, right? You can't tell that from their DNA. You might be able to tell the potential that they are more likely to get cancer because they have a trait that's connected to people who usually have cancer. But you can't definitely for sure say just from their genome, from their DNA, that they at this moment have cancer or do not have cancer. Okay, so the technologies you would use for sequencing DNA, remember, we have a sample of a person's whole DNA, and we need to sequence it. We need to see what their DNA strand is so that we can then compare it to potential other DNA strands. So one thing that is done, this isn't the way that you sequence it, but something that can be done for sequencing, if you guys remember, was gel electrophoresis. Remember, we looked at those gels and you could tell the sequence of a short set of DNA, right? You could say like it's A, T, C. You guys even had a homework problem where you had a gel and you had to figure out the sequence of a strand of DNA. Now, gel electrophoresis is not usually used for genomics because it's only really good for a shorter sequence, like something 100 to 1,000 base pairs long. And remember, for genomics, we're literally trying to sequence the whole set of someone's genes, all of their genes. So the two things that are utilized the most are what are called shotgun sequencing and high throughput sequencing. Shotgun sequencing uses something you guys are already familiar with, and that is the Sanger method, which again acts kind of similar in the type of information you get out when you read your gel. But remember, it's going to be using either a radio label or fluorescence to read out each of those base pairs that come. So you could get A, T, T, whatever, etc. Another type of sequencing technology that is used is called high throughput, and this uses, in particular, something called Illumina dye sequencing. Now, this is not one of the areas that we particularly covered, but I wanted to highlight this is one of the newer types of technologies that's being utilized for genomics. So again, the main one that you guys have talked about in how to sequence DNA is this Sanger method. And for shotgun sequencing, they're doing it at a much larger scale, but it's still the same idea, the same principle. Can any of you guys tell me why you wouldn't use PCR? A lot of you answered PCR. Does anyone know why would PCR not be good for sequencing the whole genome?
Because it can just amplify a segment of DNA, not the whole.
That's right, Hakima. Remember, the purpose of PCR is it just amplifies a segment of DNA. So you put a primer in for PCR, and it just tells you if that segment of DNA is there. It's not going to give you the sequence of the DNA. In fact, you already have to know a little portion of the sequence to just amplify that one gene per se. So if we want to collect, we want to get the whole genome, we want to know the whole sequence of DNA, we have to actually sequence the DNA using these sequencing technologies.